So far, we have talked about two mechanisms of protein transport. First one was how proteins are transported into nucleus. We said that these proteins can be fully folded and they can still be imported into the nucleus with the help of specific receptors and also nuclear specific proteins present on the nuclear pore. Then we talked about mitochondrial import. We said that there are proteins that have to be imported into the mitochondria. They have to be in a linear configuration. They cannot be folded proteins. Now let's talk about a different type of import. Now these proteins that are imported into the endoplasmic reticulum, they are imported, they're actually injected in the endoplasmic reticulum while they are being manufactured. So let's look at that. First of all, briefly, a few words about endoplasmic reticulum and smooth endoplasmic reticulum in specific. Regions of ER that lack bound ribosomes are called smooth endoplasmic reticulum. And in great majority of these cells, these regions are not plentiful. They're just specific parts of basically overall the organelle, which we call endoplasmic reticulum, mostly is rough and partly small portions of it are smooth. Smooth ER contains endoplasmic reticulum exit sites. At these sites, vesicle forms and these vesicles carry cargo in the form of soluble proteins and also proteins that are embedded in the membrane of endoplasmic reticulum, and these vesicles bud off and they fuse with the Golgi apparatus. We have seen an animation of that, and I'll show you part of that animation again. In hepatocytes, smooth ER doubles up in the surface area during detoxification of lipid-soluble drugs and various harmful compounds produced by metabolism, for example, phenobarbital. ER in most eukaryotic cells stores calcium ions, we have talked about that, which acts as a secondary messenger. In muscle cells, for example, there is abundant endoplasmic reticulum, and the endoplasmic reticulum of muscle cells is referred to as sarcoplasmic reticulum, which sequesters. One of the functions of this endoplasmic reticulum is sequestering calcium ions from the cytosol. We know if there's abundance of calcium ions in the cytoplasm, it acts as a secondary messenger and could convey different signals to the cell. Here you can see electron micrograph of rough endoplasmic reticulum. You can see that these endoplasmic reticular membranes are studded with these dark balls, and these dark balls are basically the ribosomes, which are synthesizing proteins directly attached to the ER membrane, creating regions which we call rough endoplasmic reticulum. So this is an image of electron micrograph of endoplasmic reticulum. I would like to re-emphasize that all eukaryotic cells have abundant endoplasmic reticulum, which is organized into net-like branching tubules and flattened sacs throughout the cytoplasm. Here what you're looking at is the immunofluorescence micrograph, and it is highlighting the network endoplasmic reticulum in the cell. The tubules and sacs are interconnected, so the entire endoplasmic space in a lumen is basically interconnected, and it is basically sort of a labyrinth which is inside the cell. This organelle is sort of like a labyrinth present inside the cell. This highly convoluted space is called the ER lumen or ER cisternal space. The ER membrane separates ER lumen from the cytosol, and it mediates the selective transfer of molecules between these two compartments. ER captures selected proteins from cytosol as they are being synthesized. The two types, one transmembrane proteins. These proteins are the ones which are embedded in a membrane. As you know, the receptor proteins, these are the type of proteins which are also have a domain which is present, or which has a transmembrane domain which is present in the hydrophobic part of the membrane. And also water-soluble proteins which are fully translocated across the ER membrane and are released into the ER lumen. We know in the secretory pathway, the proteins that cells secrete also are the proteins that are water-soluble, and they're injected into the lumen of ER From there, they bud into a vesicle, and then ultimately those vesicles fuse after several exchanges, fuse with the plasma membrane, releasing the contents. Some of the transmembrane proteins function in ER, but many are destined to reside in the plasma membrane, for example, receptors that we have talked about, or membrane of other organelles. Water-soluble proteins are destined either for the lumen of an organelle or also for secretion. For example, we have talked about how antibodies are produced, and this is one of the ways they would be secreted is through the Golgi apparatus route. All of these proteins have same kind of signal sequence and are translocated by similar mechanism. We'll talk about those mechanisms later, but first of all, I would like to emphasize and re-emphasize that transport is co-translational, meaning that proteins are not fully manufactured, they're partly manufactured when they bind the and as the protein is being made, it is being injected in the lumen of the So, which again contrasts with the post-translational process, which we talked about, for example, in case of mitochondria or nucleus. One end of the protein is usually translocated into as rest of the polypeptide chain is being made. The protein is never released into the cytosol and is therefore never in danger of folding up before reaching the translocator in the membrane. This is a very important part because I will explain to you later that there are certain proteins that are not required in the cytoplasm. If they're present in cytoplasm, they can be very dangerous for the cell itself. Now, I would like to show you a brief animation of this process. I have shown you previously also, but I would like to show this to you again. So I hope the animation was helpful in you comprehending how the proteins are co-translationally forced into the endoplasmic reticulum.